All right, this is an in-class lecture talking about generalized linear mixed models. I'm Jared Nemi, and we're going to be doing it in the context of a particular example that I'm calling here the Sow culling time. So if we just get right into the example, this is very wordy and not very informative, but it's from a colleague of yours. I don't know how many of you know, but Caitlin Bell is a co-PhD with statistics and animal science. And she is particularly interested in pig production process and when you should sort of retire a sow, right? So a sow is going to be giving birth to a bunch of litters of other piglets. Those piglets are going to go into the pork production process, and a sow is productive if she has lots of piglets, right? So your ideal is that you keep a sow around and as long as she's producing a high number of piglets. But you want to know when you should retire that sow and bring in a new sow for that particular pen. Right? So that's the main idea of the entire class. And so in order to build a model for how many piglets are going to be born, she provided a data set, and she comments a bunch of things. So we have this contemporary group, which is the farm year and season, which we're not going to be using for the purpose of this example, but we could use. We do know information for each litter that the sow has about which litter that was for her. This is called parity, right? So the parity is, the first parity is her first litter, second parity is the second litter. I'll probably use parity and litter interchangeably, all right? You also have information for each litter about who the sow was, and then we have information in the data set here about who the grandfather was, all right, that I'm going to be using. All right, so she has a couple of questions here, but the one that I thought was perhaps the most interesting is determining when a sow should be retired. Right, so I just tried to, all right, yeah, when should be removed from breeding. All right, we're going to go through and just show you what the data set looks like in a second, but is the main question of interest understandable, or do you have questions on that? Okay, so again, the question is, when should a sow be retired from this breeding? In terms of the parity. So basically, you have a sow, you get her pregnant, you let her have the litter, and then you have a question. Do you get her pregnant again and continue on, or do you take her out? So the question is, after each parity, do you take the sow out or keep her in? All right. In the data set, we also have number born dead. For the purpose of this example, we don't actually use that. We just figure what's important is the number that were actually born alive. But it might be of interest. You might think that the process of being dead or alive as a piglet is perhaps independent of the mother. It has something to do with the process of production or something. So, but again, we're not going to worry about that for today. Okay, so here's just a quick look at the data set. All right, so here's the first 6 lines of the data set. And we have here on the left side, this is the unique ID for the sow. Right, so sows are going to be in this data set multiple times, once for each parity that they have, or once for each litter that they have in the data set. You can see right here that this, the same sow is observations 4 and 5 in this data set, right, corresponding to the first litter and the second litter under the parity column for that sow. Right? In those litters, she had 8 piglets born alive in the first litter and then 11 in the second litter. And this is where I realized then that this is the grandsire in the sire column here, because in fact it's always the case that the sow has the same sire. So this final column is in fact the grandsire, not the sire as it's denoted here. All right, so we can look at some summary statistics here. I should have probably made parity a factor. That would have been more interesting to see how many litters 1 were in the data set versus 2 versus 3 versus 4 versus 5. You might expect that it would go down over time as sows get removed and they don't make it to those later litters. You can see that a little bit in terms of the fact that the median here is 1. Right? It's the most representative parity in the whole thing is the first litter. All right, so we can see the number born alive here ranges from 0 to 19. Right, we see that there's only 1 sow here that was actually present for all 7 litters. There's a few of them present for 6, so forth. And we see here that the sires, right, these are actually the grandsires, are represented quite often. Right, so that is one male can impregnate many females, and that's in the data set here because this sire, for instance, is in there 238 times. So he's the grandsire for 238 litters. All right, so we might be interested in how big this data set is. There's about 3,000 total observations. That's 3,000 litters. There are a total of 1,600 different sows, and there are 182 grandsires. All right, so I wanted to actually mention, before we go too much farther, that this question of interest here, right, is a question about, in the end, is going to be a question about a particular sow. Should this sow be retired or should it not be retired? All right. In the model that we're going to be constructing later, we're actually going to be using sow as a random effect. Right? But when you teach introductory mixed effect models, what you usually tell people is something like, the way that you can tell something is fixed versus random is whether you're interested in the particular unit. If you're not interested in that particular unit, you want to use a fixed effect. If you are interested in that particular unit, you want to use a random effect. Right, that's typically how we would introduce random and fixed effects and trying to get a distinction between the two. Here, we are interested in the particular sow, and yet we're going to be using a random effect for each sow. Right? And the idea here is to combine information across all the sows. Right? Treating the sows as exchangeable and putting in a prior where we learn the parameters for that prior about the sows. All right, so I just wanted to point that out before we go too much farther. Okay, data set. I think I have another. All right, questions on the data set. I'm just doing that spectacular of a job today. Or it's the exhaustion that somebody mentioned. Okay. All right. So the idea here is to build a model for the number born alive using the information we have. We have information on the parity, that is which litter, the sow, and the grandsire. All right, so that's the model that we're going to build. So here's just introducing some notation. We're going to let yi be the number born alive for the particular litter I. And then I'm going to introduce the model here in the sort of the JAGS notation. So here, pi is going to be a function that returns for each litter I which parity it was for that sow. Right? Was it the first litter for that sow, the second litter for that sow, so forth. Di, the D used to stand for dam. Now it stands for sow. So in all the notation, now you end up using D. So when you think D, you could think dam, or you could think sow. So this di function just returns which was the mother for that litter. Si returns the grandsire for that litter. Okay? All right, so then our model. Here we're going to assume now conditional. We don't see the conditioning yet, but conditional on the parity, the sow, and the grandsire. We're going to assume all the number born alive are independent. Right? They're Poisson distributed with a mean that's going to depend on the parity, sow, and grandsire. All right? We have a total of n observations here. Again, that was about 3,000. All right, so here is the function of the log of the mean. Right? Here we're calling it mu, where mu for that particular litter depends on which parity it was, what the sow effect was, and what the grandsire effect was. Right, so again, parity effect, sow effect, grandsire effect. Right, now from a Bayesian perspective, we want to treat the parity as a fixed effect and the sow and the grandsire as random effects. Right, so in order to do that, we're going to put a fixed prior on the parity. Here we're just going to use a very diffuse normal prior, right, with 0 mean and variance 1,000,000. This is basically just to be very non-informative. And then we're going to put priors on the sow and the grandsire effect that are both normally distributed. Right, with mean 0, the sows have a variance sigma squared alpha. There are total of n sows, which was about 1,600. Here's the distribution for the grandsires. Right, again, normally distributed, mean 0, variance sigma squared beta, and there are n grandsires, that was 182. Right? And now in order to actually treat them as random effects, we're going to put priors on the sigma alpha and the sigma beta, and we're just going to put some diffuse uniform priors. So again, we might refer to this as a hierarchical model, because rather than just assigning a prior for alpha and for beta, right, we're going to learn the variance terms for the alpha and the beta. So it's a hierarchical model, or it's a mixed effect model, or a multi-level model, if you will. Questions on the model? Is it clear why it's a generalized linear mixed effect model? Or should I say, is it not clear? Okay. So, all right, so generalized, right? It's generalized because it's not assuming that data are normally distributed, right? It's assuming an exponential family. In particular, it's assuming here a Poisson distribution. So that's the generalized part. The linear part. So the generalized, I guess I should say, the generalized has both an exponential family and a link function, right? So exponential family is the Poisson. The link function here is the exponential or the log, depending which way you want to look at it, right? So the link relates this term to the mean of the exponential family distribution, in this case the Poisson, right? So then the question is, all right, after we've accounted for that link function here, then we have to have everything be linear in the model, right? Because we had generalized, then we had linear. So everything here in this mu equation is linear, right? We have rho plus alpha plus beta. Right, so it's linear. It's mixed effect, which means it has both fixed effects and random effects. Here the fixed effects are rho, the parities, and the random effects are the sows and the grandsires. So because it has both fixed effects and random effects, it's a mixed effect model. Yes. So yeah, the question here is why did I choose fixed effects per parity? And the reason is, what is the good reason? I don't think these are really exchangeable, right? From a Bayesian perspective, I think that parity 1 versus parity 2 versus parity 3 are not exchangeable. That is, I have some expectation about, whether one is going to be more than the other. Now, granted, I don't really, since I'm not the subject matter expert, but I think that you wouldn't be able to treat those as exchangeable. From a non-Bayesian perspective, you might say, I really do care about what the effect is of the first versus the second versus the third versus the fourth parity, right? And so treating them as a fixed effect would make sense. Other questions? Yes.
On the right, there was a correction that said that random effect has unknown priors. Can't remember what.
Well, it should. Okay. So the question here was about the rat that the folks in class here took earlier, and it should have been, the answer was that the random effect has an unknown prior. That is, here we've assumed a normal distribution, but we have an unknown parameter in that normal distribution. For example, for alpha, the sigma squared alpha. Right. So in that sense, this is unknown relative to that for rho being known. This prior is just known; it's assumed ahead of time. This one is unknown because we're going to learn from the data what the value for sigma squared alpha should be. And since we're Bayesian, we're actually going to know the uncertainty around that value. Okay. Other questions? Okay, so the question was phrased here about predicting the random effects. And I think maybe we should say estimating the random effects, right? Because predicting would be sort of a new observation, which we're going to do in a bit, right? But the question here would be about, all right, for a particular sow, here indexed by D, for a particular sow, with the whole Bayesian machinery that we're going to do, we're going to have an estimate both for the random effect for that sow, as well as for the variability amongst all sows. So if you wanted a point estimate, we could find the posterior median or something of alpha D, but we have a whole posterior for alpha, for all the random effects, for all the sows, and for all the grandsire effects. Did that answer your question? Okay. It's going to turn out at the end of the day, when we talk about the sow culling time, we're actually going to want information both about a particular sow, but also about the variability among sows that you could possibly bring in. Right? So in the end, we're going to have a prediction problem. We are going to have a prediction problem about saying, what about a new sow? Because ultimately, this sow culling time is going to say, should we keep the sow or bring a new one in? And if we bring a new one in, we have no information about that sow, except for what we have through this distribution for the alphas. Okay. Maybe I shouldn't have gone there yet. At this point, we're just building a model to try to understand how litter size is affected by parity, sows, and grandsires. And we'll get back to that question about how to actually choose whether a sow should be retired or not. Okay, questions on the model? All right, so generalized linear mixed effect model. If we had information about the father for the litter, we would add that in. Perhaps if we had information about the grandmother for the litter, or the grandmothers or grandfathers, right, there's more of them, we could have also added that in. With the data set we have, we don't have that information. Yes? Do you always choose mean zero for the prior for the fixed effects? No, there's certainly no reason you would have to, right? It's based on prior subject matter knowledge. You might want to say that this is normally distributed around 10, because that was about the average number of individuals in each litter, right? If you put 10 in here for the mean for row with a variance of 100, it's not going to look very different than putting mean 0. So that effect is not that important. Actually, I shouldn't say 10 here, right? We want 10 on the log scale. But it's not going to make much difference what you put in for the mean for the fixed effects there was such a diffuse prior. Other questions? All right, so I think the next then is showing the model in JAGS. Now that I see it up here, maybe not the cleanest way to write the model, because I put everything into that one line for the observations. All right, so but we assume that the data here are independently distributed Poisson. The mean of the Poisson is the, right, e raised to the power here. That's the exponential function. We have in effect, rho for the parity, alpha for the sow, and beta for the grandsire. Right, and then these functions, parity I, sow I, and grandsire I, just return the parity, sow, and grandsire for that particular litter. Yep. Yep. All right, so that the top line, the yi line there, just says what the observation process is, conditional on the parity, sow, and grandsires effects. We have then fixed effects here for the parity. Right, there's only going to be 7 parities here, and they're all going to be independently normally distributed, mean 0, and precision, 1 over a million, which means the variance is a million. Right. The random effects here then have also normal distributions, mean 0, and their own precision. And then we're going to put the prior actually on the standard deviations. All right. So at least the nice thing about writing in JAGS, hopefully you figured out by now, is that it looks sort of like what you write down mathematically. Right? All right, so this is the bottom part here is just getting the data set all set up for all the information that you need for the model in JAGS. All right, any other questions on the model as written in JAGS? Clear as mud?
Clear as mud.
Good. Hopefully it's some clear mud. Okay, no questions? All right. So then we go ahead and run the analysis. Here, I chose burn-in here of 2,000 iterations. I attempted, here's the first teaching opportunity, to make 10,000 iterations total into thin by 10. It turns out in CODA samples, the argument that I wanted was thin, not n.thin, which is used for, I think, the R to winbugs functions. Right? So this did not actually do any thinning, which caused me problems with memory. All right, so ideally, I would have actually thinned here by putting thin equals 10. I wanted to comment here that this analysis took minutes to run. Right? It was not a really fast analysis. And so it's good practice then to just save the output. In this case, I used an R data file, the binary data format for R, to save the file so that if R crashed, right, I could just load it back up without having to rerun for minutes the analysis. Right? I didn't show it here, but later I calculated the quantiles. I used the summary of the MCMC object, summary of res, and I, in fact, then saved that object separately because just finding the quantiles took some time. Right? So I would get in the habit of doing that as you're coding these up. Yes?
So if it crashed, then you have to start it up again.
Yep, load. Quote. You just do load. Do quotes farm.rdata. Quotes. Yep. And then that would just load up. This save.image saves every object in R before that. You can also use what I did for the quantiles, I just used the save function. I told it to just save that quantiles object and then gave it a different file name. And then you can load that one up, and that just loads whatever it was that you actually saved. Yep. All right, so I checked this Gell-Mann, the potential scale reduction factor. I've now gone to using multivariate to not do the multivariate potential scale reduction factor. Part of the reason for that is with models of this size, it usually fails to actually do anything. And so then I just tried to look visually at all the potential scale reduction factors. Remember now that there are going to be about 1,600 random effects for the sows, so 1,600 alphas. There's going to be 182 betas. There's going to be 7 rows, and there's going to be a few other parameters, the sigmas and so forth. And so I thought I just did this manually. So this is the second teaching opportunity. Don't do this manually, right? You should just save them to something and plot a histogram or something. I thought that they were all below 1.1, and you'll see in a little bit that that wasn't true. Okay, and I'll show you how I caught that. Okay, so this runs the analysis. It should have been run for a little bit longer, given that later I find that not all the potential scale reduction factors are low enough. Again, because I did not put initial values in this JAGS.model, the initial values are just sampled from the prior, and the priors here were very diffuse. So we have an expectation that these 3 chains are sort of well dispersed relative to the posterior, so that if we do get a potential scale reduction factor that's low enough, then we're reasonably confident that at least we don't see any lack of convergence. Right? So it should be an indication that our chains have converged. All right. So the next few slides then go through some of the posterior summaries. All right, so this is the first one. This is the posterior summary for the parity. On the y-axis here are the 7 parities, right? This is the first litter for a particular sow, the second litter, third litter, fourth litter, and so forth. Plotted then on the x-axis is on the original scale of the data, right? So I've taken the row parameters here and I've exponentiated them to say, what's the expected number of piglets in each of the parities? Right, so for instance, in the first parity here, the expected number is right around 10, and our uncertainty around that number is really pretty small. Right? The expected number, right? Remember that the true number that you're going to have is a Poisson distributed random variable with this quantity as the mean, right? Which is still going to have lots of variability. But the expected number here is right around 10 with the 0.1 or 0.2 credible interval. 0.1, 0.2 being the half width of the interval. Okay, so then we see that in parity 2, it goes up. In parity 3, it goes up. Parity 4 comes down. Parity 5, it comes down. Then it seems odd that in parity 6, it goes up. And in parity 7, it goes back down. So any thoughts about what's happening? Yes?
Dealing with good sows keep producing at 6. So
Yes, that's exactly what I think is going on. And it's not even clear that that just happens at time point 6, right? That could even explain what's going on between 1, 2, and 3, right? Sows that perform poorly at time 1, they might just get rid of immediately. And so then you would expect perhaps that at time point 2, even if there's no effect of parity, you would expect at time point 2 that there would be more piglets born, because any that make it to time point 2 did well at time point 1. So what we have here is a pretty clear, I think, non-ignorable missingness. Right? So we're going to proceed in the analysis assuming that it is ignorable, but right here it seems pretty clear we have evidence that it's not ignorable. Right? And you would want to put that into your model in terms of trying to estimate these parameters for how parities are affected.
You said my comment wrong.
Oh, sorry.
No, that's no big deal.
How about these credible intervals? Why is 1 so small and 7 so big, and it seems to monotonically increase? Sample size, exactly, right? We had, remember that the median parity was 1, so we have a whole bunch of sows at parity 1. How many did we have parity 7? 1. Nice, you remembered that, right? I think that there were like 6 or 7 at parity 6. Right? So, yes, these credible intervals are exactly related just to the sample size that we have at each of those parity levels. Okay. All right. So one thing that we did not do with the fixed effect here, but we might want to consider, is having something that forces sort of a smooth function for parities 1 to 7. Right? That is, somehow you probably think that parity 2 is more similar to parities 1 and 3 than it is to 4, and than it is to 5, than it is to 6, than it is to 7. So you might want to, in the model, force that to be true. That could be done with, for example, an AR1 structure on the fixed effects for parity. Right, which seems a little bit odd. They're fixed effects, but now we're going to put a different prior that we're going to estimate parameters on. Right? Okay. But anyway, we're not going there yet. Right? That's stat 615 if you want to take it. Okay, so that's parity. The next one here is the Grandsire effect. And I note here on the y-axis that I've only taken the extremes. So there are 182 total. And I just sorted them by their medians, which are these dots. And I just took the bottom 50 and the top 50. This is why you see this very clear shift right at 50. Right? It's because the other 82 that are somewhere in between here are just missing. And this is just for visualization. If you get too many on this plot, you can't see anything. Right. Okay. So here, what do we think? So again, this is the dots are the medians. Right? Estimates for the Grandsires. The lines are then the 95% credible intervals. Yep.
Quick refresher, how to interpret this effect in terms of additional piglets born?
So yes, that's a good call. This is on the log scale.
Yeah.
So if you took the exponential of this number, it would be the multiplicative effect on the expected number born alive. Right. So here, anything that's 0, right? If you exponentiate that, you get 1. And so a 0 basically says, right, there's no effect of that Grandsire.
Since it seems to be
All right, so yeah, so it's pretty clear here that 0 is in every single one of these intervals. Right? So if you're asking about sort of statistical significance about, let's say, differences from average, right? 0 here represents the average grandsire, right? That you wouldn't be able to say that any of them are statistically significantly different from average. So what does that mean? Should we get rid of them? Should we vote? Nobody really wants to say anything. So, all right. So either we can keep the random effect in the model, or we can get rid of the random effect. So who says we should get rid of it? Who says we should keep it? And then who didn't want to vote?
I say we consult the subject matter experts.
Yeah,
Well, this point, just adding computation time. It doesn't seem to be doing anything.
it certainly adds computation time. But does it explain some of the variability in the data? It seems like it probably does. Right? So my inclination would be to keep it in. Right? Because essentially, if you take it out, you assume that there's absolutely no variability amongst the grandsires. And it seems like there probably is some variability. Yes, we don't have statistically significant results to say there are. Right? But it's my belief that there probably are differences. Yep.
Can we make anything of the fact that even though 0 is in every one of those intervals, there is kind of a pattern?
Yeah, I mean, so you could certainly say something like, ask which one is the best, right? And you can make some kind of statement about that, right? And so certainly, probably the one that's actually at top, or perhaps the one that's, no, probably the one that's at top, you're going to say is the best, right? But
Couldn't we throw the medians like a QQ plot and just show that they probably conform to
all right, so the question is, putting the medians into a QQ plot,
right?
what would be the purpose?
So just show that the effect is basically random between grandsires versus.
No. Okay. All right, so the comment was throw the medians into a QQ plot to show that the variability amongst grandsires is just random. That's fine. So it's fine. Okay, so two things. One, if you're really trying to answer the question of whether it's just random, right, you still care, right? If you have a choice of which grandsire to use, you want to use the one that's at the top and not the one that's at the bottom, right? So even though you can't say statistically that they're different, right, you still, the probability is that that one at the top is better than the one at the bottom, right? The second part is that I don't think the QQ plot really tells you that anyway. What the QQ plot might be interesting for is to think about whether your normal assumption is reasonable, right? So let's say that your QQ plot, all the data falls straight on the line. Okay, it seems like when you fit the model assuming it's normal, that in fact it looks normal at the end. But if it doesn't, right, if it seems like it has heavy tails or something, you might want to reconsider which distribution you're using there for the random effects for the grandsires in this case, right? You might want to use something that has heavier tails. You might want to use something that has heavier tails anyway, right? We have quite a lot of data here. I mean, 182 grandsires. We probably can estimate something that has heavy tails. Yes?
So there was one grandsire that was in the data set a lot more than any of the others. Do we know if that's the one at the top?
So the question was, there was one grandsire that was, I think in the data set like 240 times, which was many more than any other grandsire. So how would we identify that grandsire on this plot?
Smallest credible interval?
Right, so probably the smallest credible interval. Right, we have the most information on that grandsire. Now remember that there's 82 grandsires that are missing here. So it might be that that one is, one of the ones that's missing. But otherwise, if it is one of these sets, then yes, I would think it's likely that it's the one with the shortest interval, right? Yes?
Following up on that thought, and also the earlier plot where you were speculating that the average for the 6-letter
Okay, yeah, so there's certainly a question here about preferentially including grandsires in the data set by virtue of them being used more often to breed. I don't know how that would affect this analysis. It seems to me that you could probably set it up to be ignorable. Is that your question about whether you need to account for that?
Well, from this data, it doesn't look like it needs to be
Yeah.
accounted for here, but I would expect if like they really were on top of their breeding, that you would hope to see.
Yeah. Okay. So this is a combination of both the last 2 points, right? That you would expect that the one that was in there 240 times is likely to be one that has a large positive effect because they bothered to include it many times in the breeding. And so so you might expect, so maybe that's some rationale to say if we had to guess that fourth one down is the one that was in there 240 times. I think that seems reasonable, as long as you think that whatever they're using currently to choose grandsires is working. Other questions? Okay, so now the same plot, but now for the sows. And now there are 1,600 of these sows, and we're only doing the bottom 50 and the top 50. And so now there's a much bigger split here right at 50. Right? And I did, just for the heck of it, I plotted both the sows and the grandsires on the same axes, right? Because these are the same effects. And so if I just flash back and forth here. So one thing you might wonder about is what the effect is of grandsires versus sows. Right? And it seems like the effect is at least on the same scale. Right? These medians here fall about the same spot for both the sows and the grandsires. So, well, it's only one data point, but the one data point at the top seems a little bit like there might be heavy tails. Right? If something is normal, you probably don't expect it to be shifted that much from just the 1600th observation to the 1601 in terms of the order statistics. You can see here that relative to the grandsires, the credible intervals are almost all exactly the same length. Right? This is primarily because on many of these you only have one litter, on a few of them you have two. Right? Only one of them you have 7. It might have been interesting for me to pull out the one that had 7 just so you can see what that one looked like. Perhaps make this plot for each group of sows that had the same number parity in the data set. That'd be interesting. Okay. Thoughts on this plot? It's very similar, obviously, to the grandsire plot. Okay, so I think then I'm going to go, so these should really say now sow on the left and grandsire on the right. And these are then now the posteriors for the standard deviation parameters for the mixed effects, right? For one for the sow and one for the grandsire. All right, so what do you think? Yes? Well, I'm glad you brought that up. So the point that was brought up is and Gelman in particular points out that if the variances are near to 0, that in your MCMC you can get stuck near 0. Right? And the reason that you can get stuck is because if you estimate a variance parameter that's near 0, that means that all the random effects are basically the same. Right? And since it's normally distributed mean 0, then they're all stuck right around 0. And then you turn around and try to estimate the variance parameter, but since all the random effects are near 0, your variance is near 0. Right? And so you get stuck in this chain. And this is happening here. Why do these pictures maybe make you think that that is what's occurring? so the comment was that the distributions have mass near 0. That alone is not suggestive of getting stuck near 0. It just says that the posterior has mass near 0. Yeah, so this bimodal distribution, right? This bump here that's near 0, especially prominent for this sow labeled dam effect here, right? There's a little bit of one as well in the Grandsires, right? You can see a bump here, both of them about, 0.01 or so? Yeah, so this is the thing first that tipped me off that maybe my potential scale reduction factors weren't near 1, right? Because I might expect that these posteriors would be smooth, right? In general, you should not have that expectation, but these hierarchical models generally perform well, have a unimodal posterior, so forth. And so the fact that I saw a bimodal distribution here, it seemed like something was going wrong with the mixing of the chains. So then I went and investigated it. And so here's what we have. On the top is the plot for the sigma alpha. I guess I didn't really need the right plot since we already had the histogram on the previous page. But you can see right here that one of the 3 chains, right? So in these trace plots, the 3 different colors are the 3 different chains. And one of these chains, the red one here, did get stuck sort of near 0 or at a low value for these iterations from about, 9,000 to a little over 10,000. Right? So yes, this bimodal, this mass here at about 0.01 is definitely driven by the chain getting stuck near 0. As maybe you expect, for the sigma, for the beta, it's not quite as pronounced, but every once in a while, one of these chains seems to get stuck, right? If we run it long enough, all the chains are going to get stuck down there at some point. All right, so then I just went and calculated the potential scale reduction factors for those 2 parameters, and lo and behold, right, they're not as good as we would like them, right? So we'd like to run these chains longer to get these potential scale reduction factors lower. Right, so this took, 10 minutes or something to run. Now we're talking about, I don't know, an hour. Okay, questions on that? Yes?
What's to say it's not condensed?
So it will, right? But you need it to get stuck enough that it sort of washes out. Yeah, because all the chains should go down and get stuck there. I mean, because they're just exploring the posterior. I mean, ideally, you don't want them to get stuck. And so it might be that you can choose a different algorithm that allows them to get unstuck quicker, right? But if you're just using JAGS, then JAGS does what JAGS does. So.
Maybe this is what you were just saying, that Gelman talks about introducing, there's some Greek letter, I don't know which one it was, as a multiple trace data to unstick these.
So Gelman talks about reducing correlation in the posterior. And yes, certainly what's occurring here is a correlation between the alphas and the sigma alpha.
There's 2 things he talks about in non-convergence. One is the correlation.
Okay.
The other is when the variance is close to 0.
Oh, yes. Okay.
He has specific recommendation. I was wondering how you do that, or if you.
That's a good point. So I'd forgotten about that section where Gelman talks about, in particular, what to do with variances near 0 and adding in, I think it's an additional latent variable, right, that he adds in. He augments it somehow. Yeah, okay. So I should try that just to see, because those kinds of tricks that Gelman mentions do work in JAGS. Right. So I'll take a look at that. Other thoughts? Okay. So I just wanted to point out the analysis that you could do not in a Bayesian fashion, right? So you can do this. I mentioned that the analysis took 10 minutes or so, and this takes seconds. So it's certainly something you want to do before you even try the Bayesian analysis. I just wanted to point out that it basically coincides, right? Here you have estimates for the standard deviation for the, here labeled dam and sire for the sow and grandsires. And, right, so it's, 0.05 for the sows and 0.035 for the grandsires. So 0.05 for the sows and 0.035 for the grandsires. So it seems to match up. We have estimates here. Now this is coded differently, right? The intercept here is on the log scale. So this is the expected number of piglets in the first parity on the log scale. So if you take the exponential of this, 2.29, you get the expected number on the log scale. And that's going to be about 10, right? E to that. Then the other 6 parameters here are the difference relative to that. Right? To say, right, somewhat arbitrarily, are there differences between the first and second, first and third, first and fourth, and so forth. Right? If you figure these out, it basically matches up with the Bayesian analysis. I thought it was interesting here that I commented on my the seedling weight lecture that was online that if you just do limmer, you don't get p-values. You don't get confidence intervals. And now here, the same author of this package gives you p-values for the generalized linear mixed effect models. It's not clear to me why it would not work in the normal case and would work in other cases. So it's not clear to me why those p-values make sense or don't make sense. All right, the only other thing I wanted to point out here is that these standard deviations are just point estimates, and we don't actually have the uncertainty associated with them for the standard deviations. And that is going to come into play in what we're going to do in a second. Okay. But again, by far the rationale here to use this model is it took a few seconds to run. The other model takes minutes to run. Okay. So, all right. Okay, so now we've got a model, we've fit the model, we have some posteriors for the model parameters, but the question now is, how do we answer the question of scientific interest, right? And the real question here is, when should we remove a sow from breeding? Okay, and this question really is, who do we expect, or who do we predict will have more progeny? A current sow? Right? We're going to call this sow D. Or a new sow that you would bring in to replace that sow. Right? The idea here is, and it's sort of true, is that you have a fixed number of pens that these sows live in, and so if you take one out, you put a new one in. Right? Okay, so this is the question of interest, is to compare what we would predict for this sow versus a new sow. All right, so here's what we're looking for. The current sow, let's say, had p-1 parities, right? Had already p-1 litters. And so we're really trying to predict, now what this particular sow D, would have for its p-th parity, right? Because these just increment, right? And we're just going to, for simplification here and for comparison purposes, we're just going to stick in the average grandsire, which just means that the random effect for that sire is, or that grandsire is 0. So what we want then is the predictive distribution, and I guess I should have conditioned on right here on the left side here on the grandsire being average, right? The beta S being 0. We want the predictive distribution for how many progeny we expect for the next parity for this particular sow. Right? And so mathematically we can write that down here as the integral over the predictive distribution given the effect for that parity, the effect for that sow, and this average grandsire. Right? So this here is just a Poisson distribution, because we've conditioned on everything that we need for that Poisson distribution. And then we have to integrate over our uncertainty about the posterior for the parity and for that sow, the random effect for that sow. The spacing here is intentional to compare to what comes next. All right? All right. We're getting low on time, so I want to point out that this should give you some intuition about how to do this via simulation. Right? Via simulation, what you want to do is simulate rho and alpha from its posterior, plug that into the distribution for the Poisson distribution, and simulate a new number of progeny. And repeat that a large number of times. And then you have a full posterior for the number of progeny for that next parity for a current sow. All right, so in contrast, now we think about, we have a new sow. Well, if a new sow is coming in, it's going to be their first parity, right? And to be consistent, we're again going to use the average grandsire. And now we want the distribution for the number of progeny for this new sow. Well, we have the effect for the first parity. and we have the effect for that new sow, right? But this random effect for that new sow, we have no data on, right? Because we have not seen this sow before, and it's not in particular in our data set. And so what we need to do is we need to stick in the distribution for that sow. And I got to okay. For that new sow, right? Well, that distribution depends on the population distribution amongst all sows, right? And then we're going to integrate out over our posterior for now here the parity 1 and the variability, our uncertainty in the variability of the random effects for sows. So similarly, what we're going to do here is we're going to simulate from this posterior, which then allows us to simulate from the posterior for a new sow, plug that in, and simulate a Poisson distribution. Okay, so that's what we're going to do. Finally, then, what we're going to use is delta here is just going to be the difference in the number of progeny for the new minus the current sow. Right? So ideally what we want is delta to be big. Right? That means that bringing in new sow, not ideally. If we're going to choose a new sow if delta is greater than 0. Right? All other things being equal. Okay, so here's just rehashing what I said to do via simulation. So for the new sow, right, you have MCMC simulations from the parameters for the parity P and for the random effect for that sow. Plug them in, simulate a new realization for the next parity for that sow, for the number of piglets in the litter. Here samples a new sow random effect. And then using that, we can get a random sample from the predictive distribution for the number of piglets in the first parity for that new sow. And then finally, we're just going to calculate this quantity delta, which is the difference of the two. Okay. All right, so this delta is just then one realization from the predictive distribution for the difference in progeny. And if we take a whole collection of those, then we have a full posterior distribution for that predictive distribution, for that predictive quantity, let's say. Okay, so that's what I did here. The histogram is that distribution out of the deltas. I just tried to highlight here, not using very good technique, but where the 0 is. Right? So I made the breaks here. This difference is always going to be an integer. Right? So the breaks here are just between all the integers. And then when I looked at the histogram, I thought, man, that looks awfully normal. Right? So I went ahead and just plotted the best fitting normal curve on top of it just to see, and man, that looks normal. Okay. I can do some other things. I can say, what's the probability that delta is greater than 0? That turned out to be 50-50. And I can also say, what's the probability that it's greater than or equal to 0? Because there's actually some probability that it's exactly 0. Right? Well, that's about 0.58. We can calculate what the expected difference is. That turned out to be 0.465. And I say here plus or minus 0.03. And this right here, this plus or minus 0.03, is just the Monte Carlo uncertainty in this quantity. Right? That tells you whether you need to take more simulations to be more confident in what that is. And if all we care about is 0 here, we don't need to take any more, right? It's plus or minus 0.03. Any kind of confidence interval you want to take is going to be well away from 0. Okay, so we're running out of time. The last thing I wanted to talk about is, so we don't just make a decision based on who we expect to have more progeny, right? What we really want to take into account for is any costs that are associated with bringing in a new sow versus keeping an old sow. And so the first thing I did here, I'm just going to go through it real quickly, is to show that if the only thing that matters is the number of progeny, right? That there's no cost for bringing in a new sow or keeping an old sow, then all that really matters is our posterior expectation for the difference. And we're done. The next slide says, okay, what happens if we actually have costs for moving a new sow in, right? Maybe there's transportation costs for getting that sow from wherever it is into the production facility, right? Well, then you get this kind of formula, right? It, again, only depends on the expectation of delta, right? Plus the cost for the piglets versus the cost for bringing a new sow in. You can do the same thing. Maybe it costs more to keep the old sow because you have to medicate them more or something, right? You get the same thing here. The formula looks almost identical And then the decision will definitely depend on what these costs are, right? But the nice thing at this point is that they only depend linearly through that expectation that we have. Okay. The last example here, I say, well, that was fine because all the costs were linear in the number that we expect. But what if we don't have that? And so I give an example here where the cost function is a bit more confusing, a bit more complicated. And this ends up being trivial only because now you can simulate it. Right? You can actually simulate what this cost difference is. And this is what we get. You can find the expectation of this, for instance. Right? OK, so sorry that I rushed through the cost function stuff. That's what always happens in stats classes, right? Apparently go take an econ class if you want to hear more about that. Anyway, so this is what I tried to cover today, right? Just an implementation of a Poisson mixed effect model and how you would do it in JAGS. Thanks.